This is our class of staining method, and our course code is BMLS3001 for the course name is Systemic Bacteriology. My name is Avinash Kumar, and today we are discussing about the staining method, and our course is BSc MLT. Now we are moving toward the prerequisite or recapitulation. The recapitulation for our topic is staining is a technique used to enhance contrast in samples. Generally at the microscopic level, stains and dyes are frequently used in. Means staining is such a type of staining method or coloring method to any substance which help us to see in the microscope. Now the substance which are using for the stains are known as dyes. Dyes are made up of many synthetic substances or as well as natural substances. So for the different topic which we have, that objective of our study is to learn about the staining method. Now there are many types of staining method in our microbiology lab. So we are discussing about the different types of staining method we are using for the identification of the bacteria, virus, parasites, and different type of other microorganisms which we have in our environment. So basically, why we are learning about the staining method, because every substance has each type of properties and each function. So for every organism there is a different staining method. Introduction. Stains may use to define biological tissues, highlighting of, for example, muscle fiber, connective tissue, cell population, classifying different blood cell or organelles within individual cell. It means why we are using stain. Stains are used for the biological tissues. In our body we have biological tissues. For every organism they are having the tissue cell. What are tissue? Tissues are the group of cells, and cells are the unit cell of organism. So basically highlighting, for example, muscle fiber, we can stain muscle fiber, connective tissues, cell populations, classifying different blood cells or organelles within individual cell. What are the different type of organelles in the cell have? We have like endoplasmic reticulum, smooth endoplasmic reticulum, hard endoplasmic reticulum. Now biological staining is also used to mark cells in flow cytometry. Flow cytometry is a type of cell differentiation technique in which we can define that what are the different types of morphology of different cells, and to flag protein or nucleic acid in gel electrophoresis. Gel electrophoresis is basically a molecular technique which we are using, and staining is also an integral part of that electrophoresis. By the staining we can dye or stain different types of protein like albumin, globulin. We have different type of protein, and nucleic acid like adenosine, thymine, guanine, like all the nucleic acid, uracil in gel electrophoresis. Now different thing is light microscope are used for viewing stain samples at high magnification, typically using bright field or happy fluorescence illumination. These bright field and happy fluorescence illumination are the different types of microscope which we are using in the microbiology lab. Light microscopes are the most common microscope which we are using the any type of lab, because the source of light is bulb or sunlight. So for the staining sample we want high magnification. For this magnification we need the oil immersion. Oil emulsions we need. By the oil emulsion, uh, it increases the refractive index of the sample, and we can easily show or visualize the stain samples. Now the preparation. The preparatory steps involved depend upon the type of analysis planned. Some or all of the following procedure may be required. Wet mount. Wet mount are used to view live organisms and can be made using water and certain stains. The liquid is added to the slide before addition of organism, and cover slip is placed over the specimen in the water and stain to help contain it within the field of view. So basically the preparation of the staining method which we are using, there are many steps which involve, like types of analysis planned, which type of substance which we are using, and which type of samples we are using, like specimen, blood samples, CSF samples, and other things. Wet mounts. Wet mounts are the used to view live organism and can be made using water and certain stain. Mainly it is liquid samples. Liquid samples are using, and we can also call wet mount. The liquid is added to the slide before the addition of the organism. Like if we are taking microorganism, we can make the suspension on the slide with the help of normal slide. And a cover slip is placed over the specimen. Cover slip is a type of small cover which help to specimen to stay on the one place, and stain to help contain it within the field of view. Now after that there is a fixation, which may itself consist of several steps, aims to preserve the shape of the cells or tissue involved as much as possible. Sometimes heat fixation is used to kill, adhere, and alter the specimen so it accepts stains. Most chemical fixative, chemical causing fixation generate chemical bond between proteins and other substances within the sample and increase their rigidity. So fixation is a step in which stain and the organism is bound together or adhere together. It is the very important part, because without fixation, stain will not be able to resist on the organism, and it cannot color the organism. So before staining we have to fix a sample on the slide, and after that we have to fix the stain on the slide. So this procedure is known as fixation. Sometimes heat fixation is used to kill, adhere, and alter the specimen. Like fixation method has different types, like heat fixation, steam fixation, chemical fixation, and sometimes adheration. In adheration, we use different types of glues to fix the slide with the specimen. Most chemical fixative, chemical causing fixation, generate chemical bond. So like the when the we are using chemicals, when we are using chemical, it means it will generate chemical bond between the proteins and other substances within the sample and increasing their rigidity. Now different terms are there, like mordant. What is mordant? Now mordants are classified into two categories. The work of mordant is to make a close bond between the sample and slide, and sample and stain. So mordant are used for that. Like in the gram staining, mordant is iodine. So mordants are classified into two different categories. First is basic mordant, second is acidic mordant. Now what is basic mordant? React with the acidic dyes, example alum, ferrous sulfate, cetyl pyridinium chloride, etc. Now second is react with basic dye, example picric acid, tannic acid, etc. These are the two different types of mordant which we are using in the labs, basic and acidic mordant. Now simple staining versus differential staining. Simple staining is a technique that only uses one type of stain on a slide or at a time. Means only one type of a stain is one type of dye is using at a time, because only one stain is being used. Only one stain is we are using at the time of staining. The specimen for positive stains or background for negative stains will be one color. Therefore, simple stains are typically used for viewing only one organism per slide. Differential staining uses multiple stains per slide, means we are using more than one stains per slide. Yani, uh, if we are using a slide of peripheral smear, we are using only one type of smell, that is Leishman stain. That Leishman stain is a type of simple staining. But if we are using gram stain for the bacterial stain, it will be a differential stain, because we are using crystal violet as well as saffron and different type of sample stain also. Based on the stains being used, organisms with different properties will appear different colors, allowing for categorization of multiple specimens. Differential staining can also be used to color different organelles within one organism. So this is the basic knowledge on the staining method. In other classes you will know in the deep about the different types of method of staining and what are the different kinds of stains which we are using. So the references of this are the first, second, third, fourth. We are taking the fourth references for making this slide, and this is done for the this slide, and we will continue in the next slide. Thank you.